Wow, here we are, the very last chapter of the class. Shoo, man, you guys have put in a lot of work on this class, but you've learned a ton of stuff. Biology 212 is probably the most interesting and diverse, but the most work of any class that we have. But wow, you have earned your 4 credits. Now, let's go on to genetics. So, genetics are, you probably had a little introduction to genetics in your high school biology classes, and you might have had them in your college biology classes. So we're gonna just touch on some of this. We're gonna assume you know some, a little bit, but not much. Okay. So once again, genetics. The father of modern day genetics, Gregor Mendel, was a monk. Okay, and the monk had dedicated himself to the priesthood. And Gregor Mendel's job was to work the garden, okay? Because all the people that worked in the monastery, they had their own garden, they made their own food, they had their own kitchen, and so they had this large amount of vegetable. They had this huge vegetable garden, and Gregor Mendel was working with peas, and he was having a terrible time because sometimes they would plan on having peas for the meal, and he would open them up, and some of them wouldn't have any peas at all, or they would be little bitty peas, or they would be great big peas. So he was having a real inconsistency. So this was before we knew anything about genetics. What Gregor Mendel did that was really outstanding in science is, through the course of all the years that Gregor Mendel was there, he just continually took notes. He continually took notes. He started out and he said, okay, you know, I went to harvest the peas today and I got this and this and this. Don't know why. The next year when he went back, he said, okay, I see colored blooms. I wonder if it's these colored blooms. Then he began to watch. Then he planted different seeds. He said, well, I'll plant these peas and see if it grows this pea. So he got these great, incredible sized peas out of the pod, saved them, used them for seed, planted them, and thought, I'm going to have this incredible crop of pea plants. But instead he got a combination, and he got some that were incredibly large peas. He got some small ones, and he got some that didn't even develop. So he kept trying to figure this out. So what Gregor Mendel eventually figured out was that there was a cross-pollination there. There was a cross-pollination of the blooms, because through the years he couldn't figure it out. He saw the bees moving around. A lot of things that we know today he was discovering. He put bags over certain ones so they couldn't cross-pollinate. And make a long story short, Gregor Mendel, of course, was never famous when he lived, and then when he died, they found all this research, and no telling how long it sat there before somebody just looked through it and went, wow, this is incredible. So he's known as the father of modern day genetics. Genetics is simply what we talked about here in the last chapter. We had a zygote, the egg and the sperm combined. Okay, so in plants, it's the pollen that cross-pollinates. Okay, the stamen and the pistil. And so now what we have is we need to know what's going to happen when we have a cross between two things. So that's our genetics. So now genetics is what happens when we cross two different haploid characteristics. Okay, when half of the DNA and the other half of the DNA, we can say one from the mother and one from the father, or in a plant we can say one from plant A or plant B. So that's genetics. So we're going to skim across a couple things. I'll show you some Punnett squares. We'll talk about several things. Now, so genes, okay, when we talk about genetics, the word genes comes from genetics, all right? Genes are segments of DNA. Now, we know DNA, we talked about this earlier, is in a double helix, has the nitrogenous bases on there. So genes are segments of DNA that code for, okay, the synthesis of proteins. All right. So what are we saying there? Genes are segments of DNA that code for synthesis of proteins. Synthesis means to make, okay, and all cells are made up of proteins. So the DNA is telling that particular cell what organism it's going to be. And so when it divides, so a skin cell, when it divides, is going to continue to be skin cells. Okay. So what is the DNA? Genes are segments of DNA that code for the synthesis of protein. It tells that cell exactly what it is. Now, when we talk about chromosomes, okay, we're going to tell you 3 things about chromosomes. chromosomes are sections of genes. We say it's on the chromatid of the DNA. Okay, so when we talk about chromosomes and how many chromosomes we have, all right, we have 23 pairs or 46 chromosomes in humans, and it's different for each different organism. But what do chromosomes do? They contain DNA. All right. They are coated with histones, 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 and there's 46 chromosomes in mitotic cells, in mitosis. So 46 in mitosis. How many in meiosis? Half. Okay. Meiosis is haploid, which means half, and mitosis is diploid, which means full. So there's 46 chromosomes in mitotic cells. Okay. Now the genes now here's the thing. Now we know where the different genes are located, so we can actually unspool this DNA. Okay. Now we can actually unspool it. We can actually cause a pH change. Unspool the DNA. We can actually pull this DNA out, and we can find where the genes are for different things. So let's give you a few of them. The genes for cystic fibrosis are located on chromosome number 7. Wow! Isn't that amazing? The genes for cystic fibrosis are located on chromosome number 7. And let's give you, well, there's several here. Huntington's disease is associated with chromosome number 4. So we'll give you a couple to know there, but the main point is that we know exactly which chromosome these things are on. So let me give you those again for note-taking there. The gene for cystic fibrosis is on chromosome 7, and the gene for Huntington's disease is associated with chromosome number 4. All right, now, here's a question for you. When does a chromosome number reach 92? Okay, 92. Now, what does that mean? Okay, so at what point in mitosis does the chromosome layer actually equal 92? Well, this is right before, now you remember the steps of mitosis, we had that earlier, okay? Interphase, prophase, metaphase, anaphase, and telophase. You remember we were talking about how you could tell that. A pro enters the room, okay? In comes whoever you want the pro to be. Shaquille O'Neal enters the room, okay? So inter, pro, meta, some girl's named Anna there, inter, pro, meta, Anna, and she went and told everybody. So if that works for you, great. If not, don't be confused by that. But steps of mitosis: interphase, prophase, metaphase, anaphase, and telophase. Well, the question is, when do the chromosomes number reach 92? In other words, when have we already duplicated all the chromosomes, okay, and before we split again, and that's before prophase, okay? So when does the chromosome number reach 92? It's before prophase. Now, let's look at another example here, okay? Persons with dark skin who produce offspring with the same color have genes that are probably what? And the answer is homozygous for melatonin enzymes. Now, let's talk about that again. Person with dark skin who produce offspring with the same color have genes that are probably what? Okay, homo means same, okay? Zygous means makeup, okay? So homozygous, and then melatonin is the skin color, okay? So they're homozygous for melatonin enzymes. Now, let's talk about what we mean by homozygous and heterozygous, okay? So let's take this Punnett square, okay? So we basically, and you may have had this, you may not, I wanted to show you this. This could be for whatever traits we want them to be, all right? So this is one parent and this is the other parent, okay? So let's say that the dad is capital A, little b. What does that mean? That means that the dad has 2 traits, okay? Where did the dad get those 2 traits? From his mother and father. So it's continually carried on. Now, the dominant traits are capital, the recessive traits are lower level, okay, or lowercase. So if this is the dad, then the dad had capital A and little b. If this is the mom, it's little b, little b. Now this could have been anything. This could have been capital A, capital A, capital B, capital B. But the dominant traits are always capital, all right? Now, if it's homozygous, they're the same. So in this case, the little b would be homozygous. Homozygous, homo means same. If it's heterozygous, that means one big and one little. So these are heterozygous, okay? So we can figure the percent and chance that something will happen here. Now, while we're on this, let's stay on this, okay? So now we have genotype and phenotype. The genotype are the actual genes, okay? So the actual genes we have here, there's two different kinds. We have 50% capital A little b, that's 50% capital A little b, okay? And 50% little b. Now, it's possible we could have a Punnett square that could have 4 different kinds. It could have 25% of each one. In this case, 50% here, 50% here. So what percent do we have that is heterozygous? Okay, it's 50 because hetero means different. What percent is homozygous? 50, okay, because we have 25% there and 25% there. Homozygous means the same. Now, which one's dominant? Capital A. So that brings us into the genotype. Okay, that brings us into the genotype. So let's say that the capital A, okay, let's say the capital A represents tall and the little b represents short. All right. So what are we going to have here? Dominant always wins. So this would be tall, okay, and this one would be tall. The recessive gene doesn't show up. But if we have two recessive genes, then we get the recessive trait. So this would be 25% here and 25% here. So we'd have 50% tall, and then this would be short. So we would have 25 here and 25 here. So that would be 50% short. So if I was doing this Punnett square, we were looking at this, okay, I would have the genotype would be 50% capital A little b and 50% little b. The genotype would be capital A little b 50%, little b 50%. The phenotype, what actually shows up, the trait would be, in this case, if A was tall and B was short, would be 50% tall represented by these two and 50% short represented by these. Okay, so that being said, once again, person with dark skin who's produced offspring with the same color skin have genes that are probably what? Well, dark is dominant over light, so it's homozygous, okay, would be the same. All right? Now, the term that refers to having two different gene alleles, okay, we just gave you that, okay, two different ones is in this, that's heterozygous. Hetero means different, and so heterozygous means refers to two different gene alleles. We have an allele for A and an allele for B, okay? Now, let's talk about another one, sickle cell anemia. Sickle cell genes are present in 8% of all American population with Central American, African ancestry. Let me give you that again. Sickle cell genes are present in 8% of the American population with Central Asian ancestry. So we begin to look at that, and we know that's 8%. We begin to say, okay, so if it's 8%, is sickle cell anemia going to be a positive, is it going to be a dominant or recessive trait? And the answer is recessive. If it's dominant, then we would see it in a higher percentage of the population. But 8% of the population isn't very high, so that tells us it's a recessive trait. Okay? Now, as a result, the gene for sickle cell anemia are inherited as a form of what? And the answer is incomplete dominance. Okay? Incomplete dominance. The trait shows up, okay? Incomplete dominance means that it does not have a dominant gene. So the genes for sickle cell anemia are inherited as a form of incomplete dominance. All right? Now, let's go ahead and stop there on this one. We've got a lot more for the next video, but we've got genetics off and running. See you for the next one.